Hello everyone, today we're going to be discussing some aspects of the innate immune response, including and focusing on these two players here, the natural killer cells and macrophages, which, as we've seen in a previous video, are a type of phagocyte. So I'm going to draw them like this. But they also can secrete a lot of things, as we also mentioned in the previous video. So without further ado, when there is a virally infected cell or any type of pathogen that has antigens on its surface, actually I'll draw it here. A host cell, we'll start off with a host cell that's been virally infected, and this normally, in a healthy state, it has these antigens present on its own surface. Well, they're not really antigens, they're membrane proteins, but in infection, some of these might disappear. So this one was there, but now it's disappeared, and there might be some, like this one for example, if we make this the green one, that might be overexpressed. So we call these two ideas forced self. Actually, I'll just make a list separately on the side. So these are the triggers or the recruitment factors for NKCs. One of them is too many of these normal receptors being expressed, which is forced self. This one is loss of a normal one in healthy state, which is missing self. And we call them self because they're expressed in the healthy state by the host cells. So they're not foreign antigens, unlike what we'll see in a minute. And a third way that they can be recruited from the cell itself is by recruited I mean they just move to them and they bind to them with their complementary receptors. But they can also bind to antibodies that are bound to them because they have a receptor that's complementary to this constant region on the antibody. And that is called antibody dependent antibody-dependent cell cytotoxicity. So cytotoxicity, I'll come to explain what that means when we discuss the effects of the NKCs. So starting with, there's one more way in which they can be recruited, and that's outside of this host cell, and that is using these macrophages. So there's a relationship between NKCs and macrophages. Macrophages here release cytokines, as we mentioned, the little bombs, or I like to think of them as little bombs, but one in particular, CLCX8. Sorry, apologies, these are interleukins. So other examples are interleukin 12 and, I'll make that a 1, interleukin 15 as well. So those are involved in recruitment. So I can say in our list cytokines, interleukins from macrophages. And as I will come to in a moment, this is a mutual relationship because NKCs reinforce macrophages too among their actions. So let's move on to their actions. Why do we need NKCs at the site of infection? Well, they have two roles. Firstly, well, they have one overarching role, which is to cause the destruction of a virally infected, for example, cell, or when it's a host cell that's gone wrong. So it could be virally infected. I suppose other examples are cancer cells, or in an autoimmune response, host cells that are deemed to be dangerous. But they basically convince the cell to undergo apoptosis, and they do this with two types of molecules, perforins, which are released from the granules in the NKCs, and they make the membrane of the cell perforated with lots of little holes, through which you get the second thing, this time entering the cell, granzymes, enzymes from the granules that enter and cause the sort of breakdown of many of the organelles and damage to, so they start this cascade of damage which results in apoptosis. The other sort of auxiliary role of NKCs, this isn't kind of their function in terms of what they do in their purpose when fighting the actual disease-causing agent, but that contributes to the amplification that happens when the different cells sort of hype each other up and upregulate each other, is when the NKCs, as I mentioned, this is their relationship with the macrophages actually works both ways. So here, I'll use a different colour. They can actually activate more macrophage activity by releasing interferon gamma. So this means that there's like a bit of positive feedback. The more NKCs are recruited, activated, and have their effect, the more the macrophages are triggered, and obviously that means they can both carry out their roles in the immune response, but then this cycle can repeat as well. So that was in a nutshell the function of NKCs and macrophages and how they interact with each other, or rather how macrophages are involved in the NKC response. Thanks for listening.